Hello everyone, Mrs. Hanson. I'm excited to be here to start chapter 19. This lecture will be about lipids. Lipids, in terms of our instructional goals, really just kind of looking at the next 4 chapters as being the first of our 4 related to biochemistry. We're going to look at functional groups really in terms of biomolecules from the rest of our journey on in our course. So we're focusing in this chapter on the first of these 4 biomolecules called lipids. We'll learn how to describe the general properties of lipids, which really is difficult because there's such a broad variety of lipids. We'll learn how to classify fatty acids and describe their relationship between melting points and the number of double bonds that are contained in their structures. We'll draw the structure of wax and identify its carboxylic acid and its alcohol components. We'll draw the structure of a triacyl glycerol. Can you see that? Triacyl glycerol. And use that to describe the difference between fats and oils. Fats, of course, are solids at room temperature, oils being liquids. We can draw the hydrolysis products of triacyl glycerols to hydrolyze, to add water. You'll identify the 2 major classes of phospholipids. You might remember that word from cell membranes. Phospholipid bilayer. We'll talk about a cell membrane in this chapter. The structure of the cell membrane is many as different transport mechanisms across that membrane. A little bit of overlap with your biology classes, perhaps. We'll recognize the main structural features of steroids, such as cholesterol, and describe that relationship between your cholesterol level and cardiovascular disease. Define a hormone and list its several examples. Last chapter we talked about neurotransmitters. Here we'll identify fat-soluble vitamins and discuss those general features and biological activities of prostaglandins and leukotrienes. Leukotrienes, triple bonds, or 3 double bonds. So let's begin. Why are we studying lipids? As we said, it's the first of our 4 chapters that deal with the chemistry of biomolecules. We begin with lipids. Next chapter, we'll talk about carbohydrates, simple sugars. The third will focus on proteins and their building blocks called amino acids. And the 4th and final chapter in our semester together, Chapter 22, by the time we're done, we'll look at genetic information, comparing DNA and RNA molecules. So the first of our 4 chapters focuses on lipids. These are the organic molecules found in biological systems. Notepads ready, and we're ready to start. Chapter 19, the introduction to lipids. We know that lipids are biomolecules that are soluble in organic solvents and insoluble in water. And if I could emphasize, the insolubility in water is the quality that defines a lipid. We are looking at such a vast array of structures. When I think of every chapter so far in the organic chapter, we've talked about functional groups. We just came off of the functional groups amines. Before that, we had amides and carboxylic acids and alcohols and esters and all kinds of different functional groups. What's unique now about fats or lipids is there is no one specific functional group that defines its being. There's not one single functional group that is common and must be there to be classified as a lipid. Instead, we're going to learn that lipids are classified by a physical property of being very insoluble in water. Look at the molecule of cholesterol coming from an egg. The egg has a hydroxyl group on it. Right here is an alcohol functional group. And yet, do you notice by far the vast majority of this molecule is an inorganic it's an organic carbon chain. So lots of nonpolar carbon to carbon or carbon to hydrogen bonds. That is really the nonpolar region. So you have this really tiny area of a polar OH group, but a very large nonpolar region, making it insoluble in water. That's how we'll learn to define a fat. Look at the molecule of vitamin E being pulled from leafy green vegetables. Here I can see that we have an alcohol functional group, which is polar. And even right here is an oxygen, giving us a functional group of an ether, which is also polar. But by far, I can see a very boring carbon to carbon, carbon to hydrogen area, which dominates as the nonpolar region. That is a fat. Vitamin E is a fat-soluble vitamin. Cholesterol, a fat-soluble molecule. And now look at here. This is that triacylglycerol. Tri means 3. Acyl, we'll learn, is esters. And notice that those labeled in red. You have these kind of polar region right here where there's lots of oxygens. But you also see a large majority of just carbon to carbon or carbon to hydrogen, non-polar region, and that makes that molecule lie under the category of a fat. So there isn't one specific functional group to define a fat. Fats are defined by their physical property of being, just think about being insoluble in water. And so as we progress to the next slide, we'll define that. Lipids are biomolecules that are organic solvents. They do dissolve well in organic solvents, but they are indeed insoluble in water. And that's the definition of which we define a fat. It's based on the physical property of being water insoluble. And remember our old saying, likes dissolve likes. And so if you have a very nonpolar region in a lipid, lots of nonpolar region, and water, who happens to be very polar, remember the hydrogen, oxygen, hydrogen, this is a very polar molecule. You wouldn't expect a lipid to communicate well with a water molecule. They are opposites. So nonpolar regions in a lipid contain lots and lots of carbon to carbon and carbon to hydrogens and very few polar bonds, resulting in their water insolubility. Lipid comes from that Greek word lipos, which stands for fat. And they have such a wide variety of structures, and they have many, many different functions in our bodies as well. So let's think about that. Which compounds are likely to be lipids? Letter A is sorbitol, and letter B is beta-carotene. Now reminder, lipids contain lots of carbon to carbon or carbon to hydrogen bonds, very boring nonpolar regions, and they have to be water insoluble. So I'm just looking at this now. If I look over here, I'm noticing lots and lots of polar regions in this molecule. Notice how many OH groups we have. Those are all alcohols, and I know that those are very polar. Also, I notice I have a 6-carbon chain, and I'm remembering that solubility rule about 6 is kind of that magic number being water soluble for alcohols. So this right here would not represent a lipid. I'll just write that a few more less letters. Not a lipid. It's too polar. In other words, there's not enough carbon to carbon or carbon to hydrogen to really just overcome that polarity of the sorbitol molecule. But what about beta-carotene? Well, my goodness, I don't see any polar regions in there at all. All I see are carbon to carbon and carbon to hydrogens, which this whole molecule then is a very boring molecule. It's very nonpolar. So yes, beta-carotene would represent a structure that's classified as a lipid. We're looking for large nonpolar regions to be classified as lipids. You want to try some more? Ask yourself which compounds are likely to be lipids. Before I give you an answer, pause the video and see how well you guess. Give it a try. All right, are you back? Let's give it a try. Methylonic acid in letter A. Notice that I can point out quite a few polar regions in that particular molecule. That molecule is very polar. This is not a lipid. And when I say that, there's just not enough carbon to carbon or carbon to hydrogen bonding. When I look at letter B, menthol, there is an alcohol group. I do see an OH, but there's just one of them. So one polar region can still be classified as an alcohol. Do you remember in a previous slide when we looked at those examples of cholesterol and so forth? They had alcohol groups on them as well. So as I think about that, the carbon to carbon or carbon to hydrogen bonds is prevalent. There's lots of them. So this one could be, yes, I would classify that as a lipid. Even though there's one little polar region on the alcohol group, there is quite a bit of nonpolar region to make this water insoluble. How about letter C, estradiol? Well, I can see those two alcohol groups. There are two polar regions. However, I see a whole lot of carbon to carbon, carbon to hydrogen bonds in a nonpolar region, which is quite dominant in this structure. So yes, this would be classified as a lipid. How about ribose? Ribose is a sugar we'll talk about in ribonucleic acid in our last chapter, talking about RNA and DNA. Ribose is a sugar found in ribonucleic acid. Is it a lipid? And the answer here is no. Look at all of these exciting oxygens. There are so many polar that it's not enough to overcome the few carbons that are in there. So this one is not classified as a lipid. I do notice that in letter B, there is one polar region down here, that this is an exciting alcohol group, but I do notice also that there is a lot of non-polar region to overcome that. And so yes, this would be classified as a lipid. And pyruvic acid. Well, I can see all kinds of exciting polar regions there. Each of those oxygens creates an electron-rich area. This could not be classified as a lipid. Are you getting comfortable with recognizing what are lipids? I hope so. They're just boring molecules. The functional group can have some polarity, but we're looking really for a lot of boring non-polar region. Lipids are water insoluble. Now, we can classify lipids actually in two different categories. The first of the two is hydrolyzable lipids. Hydrolyzable. This simply means can be separated into smaller molecules by adding water. There. Hydrolyzable. There are 3 hydrolyzable lipids that I want you to memorize. I want you to remember that these are hydrolyzable, which means when I add water, I can break them down into smaller pieces. The first of those is a wax. We will be drawing waxes in section 3. We'll look at triacylglycerols in the next section, section 4, and phospholipids later on in the chapter as well. Right now, we're recognizing lipids can be classified in two different ways. We can see them as hydrolyzable, and the second will be non-hydrolyzable. And if they are hydrolyzable, I know it's a big word, but just means adding water, breaking down to smaller pieces. You can just, it's kind of like a decomposition really, but to decompose, to break down into a smaller substance. You'll notice that most hydrolyzable lipids contain that ester group. Remember the ester where you have a carbonyl, OR. And that's the ester functional group that kind of lets us see where to add water. And when we do, it's this bond that's going to break. And when we do that, you're going to get a carboxylic acid out. And on this side, you're going to get back the alcohol. A hydrolyzable, looking at an ester functional group, breaking apart the carbon to oxygen bond and breaking down into smaller pieces. We will definitely be writing that reaction multiple times. But I'm trying to emphasize the term hydrolyzable. Waxes, triacylglycerols, and phospholipids are hydrolyzable lipids. In the presence of an acid, a base, or an enzyme in our body, the carbon to oxygen bond of the ester is cleaved. And that's what I showed. Cleaved means broken. And the carboxylic acid and the alcohol are formed. And so when we just break that bond, we cleave the bond, means to break it, and then therefore we can get back out a carboxylic acid and an alcohol. And let's draw that together. Let's just take a general form for an ester, and I want you to write this with me. Here's the R leading to a carbonyl, C double bond O, leading to an O and then an R. And let's just make that R prime, just so that we know that the alkyl group could be the same or different. So here is an alkyl group, and here would just be something else of an alkyl group. In the presence of water, and I want you to think of that as water is H and OH, two separate groups, right? HOH. And in the presence of an acid, in the presence of a base, or with an enzyme, which you just define as a nature's catalyst, something that will force the reaction to occur by lowering the activation energy. There's lots of opportunities to hydrolyze a bond. And that's what this means. Adding water is hydrolysis. To add water is hydrolysis. So we're breaking apart the ester, and that bond will break right here. This is the bond that cleaves. Now, what happens? Notice that we form an alcohol, and I'm going to color code that the H from the water ends over there with the ester, the OR part, creating the alcohol. And the OH part from the water ends up here on the carboxylic acid. There's your HOH. This bond is also cleaved. So the alcohol that forms is combining these two pieces, and the carboxylic acid that forms combines the OH onto the carbon. So that color coding allows us to kind of figure out how do we get a carboxylic acid and how do we get an ester to form. And let's just repeat that one more time cleanly. Think of water as H, OH. And now when I break this bond, that's what hydrolysis does. It breaks that bond. Hydrolysis, adding water to break a bond. The OH produces the carboxylic acid, and the H from the water produces the alcohol. That's the process known as hydrolysis. Now, lipids can also be classified as non-hydrolyzable, the second category. Lipids can be hydrolyzable or non-hydrolyzable, which means adding water does not break down that molecule into simpler pieces. There are 3 non-hydrolyzable lipids. Those are called steroids, are fat-soluble vitamins, and eicosanoids. Think of like the number 8, how you have E-I-G-H-T, 8. Eicosanoids is how you pronounce that word. And that's the 3rd non-hydrolyzable lipid that you should know as not being able to be broken down by adding water. Non-hydrolyzable cannot be broken down with the addition of water. Hydrolyzable does indeed get broken down, and we saw that by breaking the CO bond, forming carboxylic acids and alcohols from an ester linkage. Well, let's think that through. In which of the solvents or solutions would a lipid be soluble? So when I think lipid, I want you to think fat, first of all, that's its common name, and I want you to just remember that the most dominating feature is that it's a non-polar, that the region of the molecule must have lots of non-polar area. They can have a little polarity, but think about it as being non-polar. And remember our likes dissolve likes rule. So letter A, we have CH2, Cl2. Now let's put that together. You have carbon in the center. It's attached to 2 hydrogen and it's attached to 2 chlorine. Dichloromethane would be its name. Now it might seem unsure if it's polar or non-polar, but remember carbon has that tetrahedral molecular geometry. Kind of think about that as the chlorines are kind of not necessarily at right angles, but they're instead, at the 109-degree bond angle of a tetrahedral molecular geometry. I say all that because all of this is very electron rich, that this molecule is very polar, pulling the electron density towards the chlorine. Since this is a polar molecule, CH2Cl2 is polar, and lipids are nonpolar, they're not going to form a solution. So lipids will not dissolve in our CH2Cl2. How about letter B? That one just says aqueous sodium chloride solution. Well, that's an ionic salt, Na plus Cl negative. That's an ionic compound. We commonly call those things salts. That's very much charged, isn't it? Since they're ions and charges, likes dissolve likes, lipids are boring, non-charged molecules. So no, lipids are not going to dissolve in salt water. And finally, in letter C, look what we have here. 1, 2, 3, 4, 5. That's just an ordinary carbon chain. Pentane, 1, 2, 3, 4, 5. Not a lot going on with that. It's a nice, boring, non-polar molecule. And that's exactly what we would want. And so, yes, letter C, lipids would definitely dissolve in the pentane solution. Likes dissolve likes. We're looking for non-polar solvents to dissolve the lipids. So in our next section on fatty acids, let's focus a little bit more on those fatty acids that are hydrolyzable, because they happen to be derivatives known as fatty acids. These waxes, triacylglycerols, and phospholipids. These are the 3 categories of lipids that are hydrolyzable. and they're coming one section at a time. But these hydrolyzable lipids are derived from fatty acids. And you're given a little taste on the front of your notepad cover of a chart, because I didn't really want to force you to memorize some fatty acids. But do you see in table 19.2 on the very front cover of your page, of your notepad page, these are some common fatty acids. And these hydrolyzable lipids, as waxes, triacylglycerols, phospholipids, they're all derivatives of those fatty acids contained on your front page. By no means is that an exhaustive list. They're just common fatty acids. Fatty acids are carboxylic acids with long chains of carbon atoms. You're looking at anywhere from 12 to 20 carbon atoms. Take a look at palmitic acid. Can you read that with me? Palmitic acid. There are 16 carbons, 32 hydrogens, and 2 oxygens. Well, that's the COO right here. That's the carboxylic acid functional group. Now, that's carbon number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 carbons in palmitic acid. You can draw that very quickly in a skeletal structure. Just go up, down, until you've created 16 carbons and end it with a COO. "16 carbons, giving us palmitic acid. Now, notice in this region here, this is kind of an exciting region, the COOH group. The COOH group is a very polar region. And since it's polar, it contains these charges, right? The oxygen is very electron rich. Here's an oxygen that's very electron rich, meaning that it's water loving. Hydrophilic means water loving. Remember how water is polar and likes dissolve likes? Water loving means that it's the polar region of the molecule. But then I noticed this really long carbon chain. This is the most boring part of the molecule. This is the nonpolar region on the molecule. And nonpolar means that it's water hating. Hydrophobic translates into water hating. You want up here? Okay, no more growling. So we have the nonpolar structure, the long carbon chains in palmitic acid that we've labeled hydrophobic. We have the COO, the carboxylic acid functional group down here, COO, COOH, which is polar, meaning that it's water loving. That's one of our examples of a fatty acid called palmitic acid. This actually contains all single bonds between the carbons, and we will learn to call that a saturated fat. Naturally occurring fatty acids, such as palmitic acid from the previous slide, always contain an even number of carbon atoms. Somewhere between carbon 12 up to carbon 20, but always with an even number of carbon atoms. You'll see the term saturated fatty acid when they have all single bonds. Another way of saying that is no double bonds in their long hydrocarbon chains. Saturated fatty acids contain no double bonds, whereas unsaturated fatty acids have at least one and sometimes many double bonds. We'll also learn that the configuration most favored in nature is the cis configuration over trans. So cis occurs in natural fatty acids, and we'll see later on in our chapter as well that if we have a trans fatty acid, it's a manufactured acid. It's made in the lab, it's synthetic. Like a tub of margarine. That's not a natural ingredient. We make it, margarine, and it's called a trans fat, which is less healthy for you than the cis, which is found in nature. Another important trend to kind of recognize is that as the number of double bonds in the fatty acid increases, the melting point decreases. So you could say that unsaturated fats typically are liquids at room temperature, meaning that we typically call those oils, and that saturated fats, which have all single bonds, tend to be solids at room temperature. And that's really what you'll call a fat. A tub of lard, for instance, Crisco are solid at room temperature. Here's an example of a saturated fatty acid. We'll just look at this together. It's called stearic acid. Stearic acid has a melting point of 71 degrees Celsius, which means that it's pretty much a liquid at room temperature. Saturated. Notice that this means in its structure, I see no double bonds anywhere between the carbons. I should make that clear because I know that this is a coup right here. Remember, fatty acids are carboxylic acid derivatives. So no double bonds at all between the carbons makes it saturated. Stearic acid contains 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons. And I did that just to remind us that we always have an even number of carbons in fatty acids. Now look at oleic acid. Oleic acid has a significantly lower melting point because it contains a carbon-to-carbon double bond, and it is in the cis conformation, meaning that the priority is coming out of the same side. 2 hydrogens up above, and then the carbon chains on the same side of the plane. So remember how we did that? We drew a plane through the double bond, and since the carbons are both going down beneath the double bond plane, we called that cis, the same side. Here's the carboxylic acid, and then we would have, well, a total of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. There are 18 carbons in this compound. But again, it's always an even number. There is another way you might hear people refer to these fatty acids as well, and it's in terms of the omega food group there, the omega vitamin groups. Omega. There. When we talk about omega, we're referring to really the number of carbon that I first hit when I'm counting to the double bond. Start from the non-polar side and just count 1, 2, 3, 4, 5, 6, 7, 8, 9. At carbon number 9, we hit the first double bond. Starting from the non-polar side, we would count 9 carbons. So another way of naming oleic acid is to call it omega-9. We'll learn a little bit more about that in upcoming practice. So here's your table that is printed for you on the front of your copy. By the way, this is a test-taking tool. It is provided for you in your quizzes, in your test, and from now on in the final exam as well. It's always in the last question if you need to click open the last question of your multiple choice quiz to find the table. So it's provided for you. You do not need to memorize. But you will have to find many of these structures in order to draw in terms of fatty acids, triacylglycerols, and so forth. We have to be able to look them up and know how to draw them. Let's just say their names so they start to become familiar. The first name is a carbon chain, all single bonds, there's 12 carbons in it, called lauric acid. 14 carbons, all single bonds, is myristic acid. 16 carbons, all single bonds, we looked at that one just a moment ago, is called palmitic acid. 18 carbons, stearic acid. And 20 carbons, arachidic acid. Arachidic acid. Good work. Now let's go to the unsaturated fats, which contain double bonds, at least one double bond, sometimes more. 16 carbons with one double bond, palmitoleic acid. Now that sounds like here. Make sure you see that distinction. Palmitic, this is palmitoleic acid. Palmitoleic acid. Here's carbon with 18 in the chain, one double bond, and that's oleic acid we just looked at. 18 carbons but with two double bonds is linoleic acid. Linoleic acid. 18 with with three double bonds, linolenic acid. And 20 carbons with a double bond right there, you have arachidonoic acid. These are fun to say. As I said, I'm not making you memorize, but the more you hear them, the more you'll recognize those as fatty acid names and know then to pull out your tool chart to be able to write them. Linoleic acid is called a omega-6 acid because of the position of the first carbon-to-carbon double bond, and that's kind of what we had said a moment ago. Linoleic acid is an omega-6. When I count from the nonpolar side, at the nonpolar side, start there, you count 1, 2, 3, 4, 5, 6. The site of the double bond, the first, I know there's at least two in this structure, but the first one you hit, that's how you would name it, as an omega-6 acid. Linolenic acid, now hear the difference in name, linoleic, linolenic. It contains the first double bond between carbon 3 and 4 right here. So it's called an omega-3 acid. The position of the first double bond in the nonpolar chain is how you hear the omega part in the nomenclature for these fatty acids. Let's kind of think through a little bit. We're asked to draw a structure of gadoleic, I got that right. This is gadoleic acid. It's got 20 carbons in it, and it's obtained from fish oils. All right, 20 carbons. So you know what I noticed? I have a methyl, then 9 more. That's adding to 1, that's 10. On the 11th carbon, I have a double bond. So what I'm going to do is just start with that double bond and know that that's going to be between carbon 11 and carbon 12. And the reason I'm doing that is because I know that in nature, the cis conformation is necessary. So I just wanted to make sure if I'm obtaining this fatty acid from a fish oils contain cis configurations. Those are definitely a natural omega acid. Between carbon 11 and 12, I'm noticing that we have a double bond. So then let's just put the rest together. This would be carbon 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Notice I kind of just made a staircase. That's really how the bonding will go. Let's verify I did that right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And there's carbon 11 to 12. Now back to the other side. We have to do a total of 20 carbons. So here's carbon 13 to 14, 15, 16, 17, 18, 19, 20, And on 20, you have your carboxylic acid functional group. So just to emphasize, we went to carbon 11, 12, where the cis configuration is. Then we did 13, 14, 15, 16, 17, 18, 19, 20. And there's the Cu functional group. We drew a skeleton structure. I suppose I should have went like this. 1, 2, you know what? That might be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then over to the cis conformation, and then back down. So that's kind of thinking about Cu down here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and between 11 and 12 was my double bond. 13, 14, 15, 16, 17, 18, 19, 20. That would be the skeletal structure. Kind of looks like a staircase. Label the hydrophobic and hydrophilic portions. Well, right here is the only exciting portion on that molecule. The Cu is the polar region. Therefore, this would be the hydrophilic, which means loving. And how about the hydrophobic? Well, that's this part over here, isn't it? The entire structure here is hydrophobic. It's water hating. So clearly I could see this classified as a lipid. It has a tiny little polar region, but dominated by the nonpolar carbon tail. Predict how its melting point would compare to the melting points of these other acids, arachidic acid and arachidonic acid. Take your time. Arachidonic acid, arachidic acid. Well, the first one here, arachidic, is saturated. In other words, it has no double bonds. And this one here, notice this has a group of 4, so it's got 4 double bonds. And I learned that the more and more double bonds, the lower the melting point. So we know that unsaturated means a lower melting point. And the more and more double bonds you have, the lower the melting point is the trend. So if we have, kind of thinking about this through, this would have the highest melting point because it has no double bonds. The one that we just drew would be in the middle because it has 1 double bond. And the lowest melting point would be the one with the 4 double bonds. So this would be the lowest between the 3. And I have that staring at us right here, thinking about the cis conformation of the hydrophobic, hydrophilic end. And we can see that one cis would give its lower melting point than the completely saturated. That's the saturated all single bonds. But certainly in the middle there, because if you have 4 double bonds, that by far would have the lowest melting point of all. And that's what we had just stated. How about you try one on your own to see how you're doing with this lesson? A little more practice. Pause the video, work it out, and see what you find. Draw a skeletal structure for each fatty acid. Label the hydrophobic, hydrophilic ends. Without referring to the table, which fatty acid has the higher melting point, and be able to explain your choice. Give it a try. Well, here I know I can see 18 carbons altogether. I know that fatty acids always have an even number of carbons. So as I just think about straight chain, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. There's my 18th carbon. So that turns into my there's my COOH functional group. This area is the polar region, meaning that it is hydrophilic, water loving. And this region, just everything else, is the hydrophobic, water hating. And now let's compare that to this carbon chain. Well, I got to do some counting here, don't I? There's 1 plus 4. And then on the 5th carbon, I have a double bond. And then I'm going to just kind of draw it here. Here's carbon to carbon, and I just want to make sure I can see those as cis, right? And every time I see this, it's cis conformation. So you just want to make sure you're coming out and drawing that in the same conformation. 1, and then 4 more. 1, 2, 3, 4. All right, so that's carbon 1, 2, 3, 4. I need one more because this double bond is going to be between carbon 5 and 6. And I want you to just make that straight across, and that's going to be your cis conformation. And then we'll come back up, and there's your carbon 7. Now that's carbon 7. 1 plus 4 is 5. This is at carbon 6 to 7. Oh, let me start over. My bad. I got to count with you. There's no shortcuts. Keep checking, checking, checking. Carbon 1 plus 4 more is a total of 5. Then on carbon 6, we see the double bond. Let me just repair that quick. 1, 2, 3, 4, 5, 6. There's my double bond. Between carbon 6 and 7 is the double bond. And so that's where I'm going to make the cis conformation. And now I can come out and find carbon 8. That was 7. That's 8. Here's carbon 9. And there's another between 9 and 10. That would be another conformation where we have a double bond. So I go back up and have another double bond. And again, the cis conformation. Now how many are left? 7 more plus the N, so 8 more. 1, 2, 3, 4, 5, 6, 7, 8. And then finally your Q. So down here would be C double bond O, OH. Oh, I got rid of that. I'm just trying to draw an OH here, but I've run out of room on my screen. Yeah. So with that particular structure, here it is kind of drawn clean for you. A cis conformation where I've numbered all 18 carbons and have drawn all saturated carbons here. So this is what we would refer to as saturated. And this is our unsaturated. We had a double bond between carbon 6 and 7. We had a double bond between carbon 9 and 10. And we can see that they're both in the cis conformation. Now thinking about labeling, as we drew those, remember that it asked for hydrophobic, hydrophilic on some of those as well. We know that the COOH groups are hydrophilic, polar, water-loving, and that the non-polar chains are hydrophobic, water hating. We would see that this has 2 double bonds, making it an unsaturated hydrocarbon, meaning that it has a lower melting point as compared to this saturated. And the saturated itself means that it has a higher melting point. How did you do? That just takes some practice, doesn't it? If we think just a little bit more, give the omega N designation for oleic acid and arachidonic acid. Now I put this chart on here and you'll be provided for it on your test, but just know that this is something you wouldn't have to memorize, but you have to be familiar with where to go find the answer. Oleic acid. Well, I can see oleic acid right down here on my table 19.2. What I want to know is, where do you see the first double bond starting from the non-polar side? So start here and count. And you'll do the same for this acid, arachidonic acid. Start at this end and count. Where do you see the first double bond? At what carbon? And that will tell you the omega. Now write them out. Take a minute and write down your answer before I share. Well, maybe you've just peeked. Here in our example, the omega N designation for oleic acid occurs at carbon 9. Here's 1 plus 7 is 8, and here's carbon 9. Between carbon 9 and 10 is our double bond, so you refer to that as omega 9 acid. Here's carbon 1 plus 4 more gets you to 5. So at carbon 6 is our first double bond. So you refer to that as an omega 6 acid. We have done great work starting out with our chapter on lipids. In our next video lesson, we'll talk about waxes in section 3. Take a little break. Come on back when ready.